So glycogen is also known as what? Animal starch, because the structure is similar to the amylopectin but more highly branched. Amino acids are nothing but a protein where it consists of NH2 group as well as COOH group. So based on the chemical nature, we can classify the amino acid into three types. The first one is acetic amino acid, basic amino acid and neutral amino acid. Hello my dear students, a warm welcome you all. Myself is Purnima, lecturer in Department of Chemistry at Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. My dear students, in our last discussion, we were dealing with a unit called Biomolecules. Under that, we have discussed about the cyclic structure of glucose and we also discussed about the disaccharide. Under that, we have discussed about the maltose and lactose, and also we have discussed about the polysaccharide. In that, we have discussed about the starch. Today, let us move on to the next topic of this unit about the polysaccharide. The remaining, which are the polysaccharide which is there, we are going to understand in this session, and we are going to understand the importance of carbohydrates in our daily life and amino acids, classification of amino acid, and mainly the Zwitter ion formation in the amino acid we are going to understand. Okay, let's see what is the another example for the polysaccharide. The another example for the polysaccharide is cellulose. Already we have learnt about the starch. Is it right? Let's see what is the cellulose, what is the structure of the cellulose. Let us understand. So already we know that cellulose are extremely found in the plant cell. Is it right? It is exclusively found in the plants and they are mainly found on the cell wall of the plant cell. Especially in the plants, especially on the cells, on the cell wall, we can see that the cellulose will be present in the green plants. And the cellulose are the straight chain polysaccharide composed of only beta-D glucose unit, which is composed of what? Only the beta-D glucose, and which are joined together by a glycosidic linkage between C1 of the one glucose unit and C4 of the another next unit. Here you can see that this is a 1 carbon, 2, 3, 4 and 5. Is it right? Yes. So this is a 1 glucose unit and you can see that 1, 2, 3, 4 and 5 and 6. This is 5 and this is 6. You can see that these two are the glucose unit and they are linked together at a carbon 1 and at a carbon 4 by forming a glycosidic linkage, and it will be known as cellulose. So where does it present? Already we have understood, especially in the green plant on the cell wall. That is in the cell, inside that you can see that there is a wall. In that you can see that this is a green portion. You can see that this is the outermost layer in the cell, that is known as cell wall. In that you can see that the cell wall, in between the cell wall you can see that there is a formation of fibrous cellulose. It is present. If you have taken out any one of the macrofibrils, you can see that how this cellulose chain will be present. It will be in the form of joint structure where each glucose unit are joined together by a glycosidic linkage. Especially at the carbon 1 to the carbon 4, they are linked together and forms a glycosidic linkage. So hope you are understood with the cellulose. We'll move on to the next polysaccharide, glycogen. So already we have understood that the cellulose which is present in the plant cell, or you can say that it is exclusively present only in the plants, whereas glycogen which is present only in the animals. Okay, it is stored mainly in the animal bodies as a glycogen, and it is also known as animal starch. It is very important. So glycogen is also known as what? Animal starch, because the structure is similar to the amylopectin but more highly branched. If you Look at the structure of amylopectin, which we have already learned in case of starch, and also with the structure of the glycogen. They are almost similar, but there is a much branching we can see in the glycogen. Hence, we can say that it is a animal starch. And these are present in the liver, muscles, and in the brain. And when the human body needs a glucose, the enzyme which breaks the glycogen and into glucose. So what if the body need energy? What will happen? Our body itself will store this glycogen, especially at the liver, as well as at the muscle, as well as in our brain. When a body needs the energy, any one of the enzyme which is found in our body can break down this glycogen and give rise to the energy in the form of glucose. So this is another one of the point which highlight for the glycogen. And this glycogen which is also found in the yeast as well as in the fungi. Not only in the animal body, we can also found in the microorganism also. Here I have a picture of glycogen. You can see that the glycogen here also, they are bonded together by a glycosidic linkage. The carbon, so carbon 1, and you can see that at carbon 4, 1, 2, 3, 4, 5 and 6. Between the first carbon atom and at the sixth carbon atom, you can see that there is a formation of glycosidic linkage in case of glycogen also. But when there is a branching, you can see that branching will be occurs at carbon 1 to the carbon 6. Is it right? This is the first, second, third, fourth, fifth, and this is a sixth carbon atom. The sixth carbon atom as well as the first carbon atom, they link together by forming a glycosidic linkage. This lead into the branching in glycogen. So hope you all understood that what is the glycogen, how we can show the Howard structure for the glycogen. Hope you all understood. Remember, glycogen is a animal starch where it is found only in the animal as well as in the microorganism. Whereas our cellulose, which is found mainly in the plants. Okay. Next comes to the important uses of the or importance of the carbohydrate. Already we have learned that carbohydrates are very important for the human life. Is it right? Which play a very important role just by providing the energy to do all the works in our daily life. Let's see the importance now. The first importance says that the carbohydrate is essential for both plants and animals. Is it right? In order to cover the each cell, we need a cell wall. Is it right? In the plant cell, that itself will be made up of the cellulose. And not only for the plants it gives rise to the energy, in case of the animals also it provides a lot of energy in the form of glucose. Then the honey is being used for a longer time as an instant source of energy in Vedas, especially in the Ayurvedic system. So you would have seen that in making the Ayurvedic medicines and all, they'll use the honey as one of the basic source for their preparation. So it will be another good example for the sources of carbohydrate, which is also used in Ayurvedic medicine for the preparations of the medicines. Then carbohydrate will be used as a storage molecules as a starch in case of plants and glycogen in case of animal. So just now we have understood that the plants will store this glycogen especially on the cell wall. Is it right? In the form of cellulose. Whereas animal can also store this carbohydrate in the form of energy molecule, especially at the liver, at the muscle, as well as at the brain. When the bodies need energy, it will be converted into a glucose and it will be provided to the particular site. So that is another important use of the carbohydrate. The cell wall of the bacteria and the plants are made up of the cellulose, and they provide the raw material for the many important industry such as textile, paper, liquor, as well as with the breweries. Not only it helps us to give energy, it also acts as a raw material in the paper industry as well as in the cloth industry. It is a main basic sources. Here I have a picture. You can see that whatever the food we take, which is a basic source of carbohydrate, which is in the form of sugar, starch, or in the form of fibers. Is it right? When the person would take the different sources of carbohydrate, it will be digested in the body and this glucose will be converted into glycogen, and when the energy is needed, this glycogen is converted into glucose and it will be given to the particular site of that parts. Okay, this is how the carbohydrate, which play a very important role, especially in the plant as well as in the human being. Hope you all understood this importance. Next, we'll go for the amino acids. Already we have learned that amino acids are nothing but a protein where it consists of NH2 group as well as COOH group. Is it right? So, which is a functional group which is present in the amino acid? There are two different functional groups are there. The first one is amino group, that is NH2 group, and COOH group, that is carboxyl group. These two different groups are attached to the amino acids. When you take the protein, if you do an hydrolysis, you will get the amino acid. And remember, there are 20 different types of amino acid will be synthesized in the human body. So, you will understand that which are all the important amino acids which will be synthesized. Okay. Let's see the definition now. Amino acids are the amino and the carboxylic functional group, and only alpha amino acid they obtained by hydrolysis of protein. And remember, our amino acids are crystalline solids and usually they are colorless. Okay, and these are water soluble and high melting salts or behave like a salts rather than the simple amines and carboxylic acid. So, we have understood that it is a crystalline solid, it is always soluble in water. Even though it consists of amine group as well as with the COOH group, that is a carboxylic group, it behaves like a common salt. It behaves like what? Common inorganic salt. That is another speciality of the amine. And this behavior is mainly due to the presence of acidic and a basic group. That is very important. So, here I have a picture of amino acid so that you will understand that which is a basic group and which is a acidic group here. So, you can see that this is a general representation of the amino acid. You can see that there is a C double bond OH. This group is a carboxylic group and this is a acidic group. This is a acidic group which is found in the amino acid, and next to this carbonyl group you can see that there is a carbon. After this you can see that there is a NH2 group will be there, and this is a basic group. This is a basic group which is found in the amino acid. In between this one basic group and one acidic group, you can see that there is a carbon which is there, and this is known as alpha carbon atom. This is known as what? Alpha carbon. Why is it so? Because it is present next to the carbonyl carbon atom. Already in the aldehydes and the ketone, we have understood that what is a alpha carbon atom and what is a alpha hydrogen atom. Is it right? Same here. You can see that there is a carbonyl group will be there. Next to this, there is a one alpha carbon atom will be there, and there is a one alpha hydrogen atom will be there. If you can see that below this, there is a one R I have written. This is a variable group which is present in the amino acid. This is how we can easily understand which one is the acidic group and which one is a basic group which is present in our amino acid. Because of these two groups which is present within the amino acid, even though it present, it behave like a common salts. Amino acids are generally represented by the 3-letter symbol and sometime with the 1-letter symbol also. The structure of the sum of the commonly occurring amino acids, I'll show with the example. Always remember, we can represent this amino acid with the 3-letter and we can also represent with the 1-letter. I'll show you with an example so that you will understand here. So here I have listed out the 20 different types of amino acid. So you will understand that how to name it and how to represent it. So here the name which I have given, and characteristics features of only R. So already I told that in my structure only R group is variable. The R group may be H, CH3, CH3, C2H5 or NH3 like that. Okay. So if R group variable, only I have written the R group, and what is the 3 letters and what is the 1 letter? Let's see now. First we'll go for the first amino acid, that is glycine, and variable group is H, so that I can write the structure of the amino acid as C double bond O, OH, H, and variable group is H now, then NH2. So this is the structure of the glycine, where my R is substituted with a H, and the 3 letters name is GLY. The first letter of the amino acid, that is GLY, will be taken as the 3 letter name, and in that you can take the first letter as a capital letter and it will be named as 1 letter code as G. Next we'll go for the alanine, where my variable group is CH3. So I can write the structure as NH2H, CH3, COOH. So this is a R which has been replaced with the CH3, and the first 3 letters for alanine is ALA. So it is ALA and the capital letter is A. Next I'll go for valine, which is very important. Okay, valine, and the variable group is CH3 to CH, and the first 3 letter is VAL and the capital letter will be V. Next is leucine, and the variable group will be CH3 twice CH and CH2, and the first 3 letter will be taken as a 3 letter word LEU, and the capital letter L is a leucine. Then isoleucine, again this is also important. This is also very important for your examination point of view. Variable group is CH3, CH2, CH3 and CH, and the variable group is IE. After I, so you can see that LE will be there. First I have taken out the I, and next after I, so LE will be there. So it is IIE, and the capital I is a letter that used to indicate this isoleucine amino acid. Next is arginine. The variable group is CH3, CH2, NH2, CH2 thrice, and the first 3 letter will be the 3 letter symbol, and capital R will be used for arginine. Why we are not using A? Because already we have used the A for alanine. Okay. Next is lysine. Again this is another important amino acid. The variable group is NH2, CH2 4 times, and LYS is a 3 letter word, and capital K is used for lysine. We are not using L because already L is used for leucine. Next is glutamic acid. The variable group is NH2, C double bond O, CH2, and first 3 letter is a 3 letter word, and capital E will be used. We cannot use the G once again because the G is already used for glycine. Then comes to the aspartic acid. The variable group is COOH, CH2 and CH2. 3 letter word is ASP, and 1 letter code will be D. Because we cannot use the A because already we have used for alanine. Okay. Next comes to the glutamine. The variable group will be COOH and CH2, and the variable word will be GLU, and it is considered as a Q for 1 letter code. This is a 10 important amino acid I have listed now. Let's see with the remaining. As per gene, group are NH2, C double bond O, CH2 and CH2, and the 3 letter word is ASN. We cannot take it as ASP, so I have taken out the ASN which is there here, and the capital letter N will be used for this. Then for 309, the variable group will be CH3CHOH and it will be represented as THR for the 3-letter word and for the 1 letter we have used T. See, right? And the variable group will be CH2OH and it will be represented as SER and the 1-letter code will be S. Similarly for cysteine. So here in the cysteine as well as with the serine, you can see that the CH2HS group will be there and here only OH group will be there. So for your competitive exam, they'll ask you which amino acid which having a variable group which consists of OH. There is a hydroxyl group. So you can name that amino acid as a serine. Okay. Let's come to the cysteine. Here the variable group will be HS and CH2 and it is 3-letter word will be CYS and capital C will be used for 1-letter code. Similarly for methanoin, the variable group will be CH3S, CH2, CH2. You can see that both in methanoin as well as in a cysteine, the sulfide group will be there. So that is a common thing that you can easily remember it. And here the first 3 letter will be considered as a 3-letter code and capital M will be used for 1-letter code. Then comes to the phenylaniline. So it is C6H5CH2 and the first 3 letters will be goes like this, that is PHE and capital F will be used here. Next is threosin and the variable group will be goes like this, that is C6H4CH2OH and first 3 letters, that is THR I have used because THY we cannot use, somewhere we have already used and the capital Y will be used for 1-letter code. The remaining is tryptophan. Again it is very important amino acid which are having variable group. You can see that there is a 6-membered ring and there is a 1 5-membered ring which are having a nitrogen as well as with a CH2 and first T, R and P I have considered for tryptophan and W is a 1-letter code for that. Then histidine, the variable group will be having 5-membered ring having NH, N and CH2 as their group and histidine for that first 3 letters I have considered, that is HIS and capital H will be used for 1-letter code. The last one, proline, here you can see that there is a different functional group that you can identify which is a 5-membered ring. I can rewrite this NH, CH, CH2 and these 2 groups also it is CH2, CH2, CH2 and this carbon atom will be having COOH. Okay. So this is a variable group which is present in proline and first 3 words will be used for 3-letter word, that is pro and capital P will be used to represent for the proline amino acid. So this is how we can represent the amino acids with a variable group along with the 3-letter words as well as with a 1-letter code. So hope you are understood with this naming. We'll move on to the classification of amino acid now. We can classify the amino acid into 3 different types based upon the chemical nature. So based on the chemical nature, we can classify the amino acid into 3 types. The first one is acidic amino acid, basic amino acid and neutral amino acid. So what are those? Neutral amino acid, basic amino acid and acidic amino acid. It is mainly based upon the chemical nature. Okay. Again, we can classify the amino acid into 2 types based upon the dietary requirement. In what amount or in what requirement the body is needed this amino acid. Based on this also, we can classify the amino acid into 2 types. They are essential amino acid and non-essential amino acid. So, this based upon the dietary requirement, we have classed the amino acid into 2 types. They are essential and non-essential. This class of classifying the amino acid, it is very important for the examination point of view. Let's see what is the neutral, acidic and basic amino acid. First, I'll go with the neutral amino acid. If I say it is a neutral amino acid, in such kind of amino acid, we know that there will be a one NH2 group and one COOH group. Both acidic group and basic group will be there. If acidic group and basic groups are equal, then we say it is a neutral amino acid. The best example is glycine. So I can write the structure as CH, COOH and NH2. So there are only one NH2 group will be there and only one COOH group will be there. So that acidic group is one and one basic group will be there. Hence it is an example for the neutral amino acid. Whereas acidic amino acid, in case of acidic amino acid, acidic groups are more than compared to the basic group. The best example is aspartic acid. So the best example is aspartic acid, and I can write the structure as CH, COOH and NH2, CH2, COOH. We'll check this structure with the aspartic acid. You can see that COOH, CH2 and CH2. This is a variable group. Is it right? CH2, COOH. In this example of aspartic acid, you can see that there are two acidic groups are there. There are two acidic groups are there, but there is only one basic groups are there. Hence, it is an example for acidic amino acid where the acidic groups are more. If I comes to the basic amino acid, the basic group must be more than the acidic group. Example is lysine. So I can write the structure for the lysine also. CH, C double bond O, OH, NH2 and variable group is CH2, NH2. I'll go with the structure now. So NH2, CH2, 4 times it is. It is 4 times. So in this example, you can see that there are 2 basic groups are there. How many basic groups are there here? 2. Whereas the acidic group, there are only 1 acidic groups are there. Since my basic groups are more, hence it is named as basic amino acid. So hope you all understood with this example where I have classified this amino acid into 3 types only based upon the chemical nature. Now let us see there is another class of amino acid based upon the dietary requirement. One is essential amino acid and the one is non-essential amino acid. If I say it is an essential amino acid, remember this amino acid will be required for our body, but we cannot prepare this amino acid inside our body. Such amino acids are taken through the diet. Such amino acids are said to be essential amino acid. Example is valine and leucine. Whereas non-essential amino acid, these are the amino acid which will be synthesized or produced within our body, so need not to taken through the diet. Such amino acids are known as non-essential amino acid. The best example is lysine and alanine. So, hope you understood with the other 2 classification. Remember, these 2 classification of amino acid, it is very, very important for the examination point of view. They can ask you to write for 2 marks. Next, we'll go for the zwitter ion formation in the amino acid. So, what is that new concept? Zwitter ion means it is a dipolar ion which is found in amino acid. Di means what? Dipolar ion means what? There will be a one positive charge and there will be a one negative charge. However, amino acid which forms that positive charge and the negative charge, let's see now. Here, I have taken out the example of amino acid. We know that our nitrogen, which is one of the group, which is a basic group, which is present in amino acid. Whereas COOH group, which is a acidic group, which is present in our amino acid. We all know that the nitrogen which carries a lone pair of electron, hence it will be requires one more electron to become a stable. Is it right? To form a ammonia. You can remember it. so that the COOH from the amino acid, that is the acidic group, which loses one of the proton, and this proton will be accepted by the acidic group to form a NH3 and COO minus. This formation of NH3 and COO minus in the amino acid, which is known as Zwitterion formation, where you can see that there is a possibility of two different ions, that is both cations and anions will be formed. How does it forms? Only because of the loss of one proton and gain of one proton. Where does it occurs? Especially in the aqueous solution. So in the aqueous solution of amino acids, especially the carbonyl group, I can write this reaction as CH NH2 R and C double bond OH. So this carbonyl group which are having the one of the hydrogen, it will be loose, so that this hydrogen will be accepted by this nitrogen and forms what? CH R NH3 and C double bond O minus. So where H will be carries plus charge. So you can see that there is one positive charge will be there and one negative charge on the oxygen. So there is a dipolar ions which is formed in an aqueous solution. Such dipolar ions which is formed in the amino acid, that is said to be Zwitterion. That is said to be Zwitterions. So hope you all understood with the Zwitterion formation, and again it is the most hot questions which will be asked repeatedly for the 2 marks. So they can ask you to write what is the Zwitterion formation in the amino acid, give an example. So you can write with your own words and you have to represent this reaction. Okay. Let's move on to the peptide linkage. So we know that amino acids, it is nothing but a protein, is it right? And it will be having a dipolar ions also when it is placed in aqueous solution. But how this peptide bond will be formed? If I consider any 2 amino acids, each amino acid we know that which consists of one carboxylic group and there is a one amino group. If I consider one amino acid, here you can see that I have a glycine. And also I can consider another one amino acid where it consists of NH2 group. If I consider 2 different amino acids where at one side COH group is there and another side NH2 group will be there. If I combine these 2 amino acids, there will be a elimination of water molecule will be taking place. If water will be gets eliminated, it will be form a link between NH2, CH2, CO, NH. So, between the CO and NH, there is a bond which is formed and this is known as peptide bond. How does this peptide bond will be formed? Between the 2 amino acids, where one amino acid will be having COH group, while another amino acid having the NH2 group, where the elimination of the water molecules will be taking place and results in a formation of peptide bond. So, if 2 amino acids are there, then it results in a formation of 1 peptide bond. If 3 amino acids are there, then results in a formation of 2 peptide bond. If number of amino acids are more than 10, then the products are known as polypeptides. If more than 10 amino acids are there, then we say it as polypeptide. This is how we can predict the bonding between the 2 amino acids in the elimination of water molecule, which results in a formation of peptide bond. Again, this peptide bond linkage, how does it forms between the 2 amino acids? It is very important for the examination point of view. Have a look on it and try to write one example for that to show that how the peptide bond will be formed. So, hope you understood with this session. In the next session, we are going to discuss with the protein and its classification. By that, I like to say that what are all the things which I covered in this session. Again, it is very important for the examination point of view. Wherever you are not understanding, please pause the video and try to note down. We'll meet you in the next session. Till that, take care, be safe. Thank you.